Hello everyone, I'm Dr. Mary Newport. I'm grateful for the opportunity to give this video presentation of my paper on ketogenic strategies for Alzheimer's disease and other memory impairments, which was published in April 2024 in Medical Research Archives, the Journal of the European Medical Society. Here is an overview of what we will discuss in this presentation. Age is the greatest risk factor for Alzheimer's, which affects about 1/3 of people over age 85. The greatest genetic risk factor is carrying the ApoE4 gene, which is present in about 25% of the population, but 40% of people with Alzheimer's. Having one ApoE4 gene increases the risk of Alzheimer's from about 5% to 12 to 15%, but two genes increases the risk to 40% with a greater likelihood of early onset. Dominantly inherited genes account for less than 2% of cases, and people with Down syndrome have a high risk of developing Alzheimer's by middle age. Having diabetes carries a 30% risk of developing dementia. Experts now say that 30 to 40% of cases of Alzheimer's and dementia might be preventable through modifying lifestyle risk factors, with diet and exercise at the top of the list, and therefore consuming a whole food diet, such as a Mediterranean diet, could be beneficial. Humans have trillions of neurons and other types of brain cells called glia that protect, nourish, and maintain the neurons and nerves. Each of these types of cells is affected in Alzheimer's disease, but can also be affected during the aging process to a lesser degree. The classic hallmarks of Alzheimer's disease are plaques and tangles in the brain, which were first described by Dr. Alois Alzheimer in 1906. Dr. Alzheimer also described abnormal lipid droplets, which have only been studied for about 10 years. Soluble beta amyloid is a beneficial substance. It is known to be antimicrobial, but beta amyloid peptides can misfold and aggregate into plaques, which can impinge on cells, nerves, synapses, and blood vessels. The plaques tend to appear in the brain in elderly people, but in general, these plaques are more abundant in people with Alzheimer's disease, although this is not always the case. Some people with large amounts of plaque in the brain have normal cognition. In Alzheimer's disease and in other types of neurodegenerative diseases, neurofibrillary tangles also develop in the brain. These come about from hyperphosphorylation of tau protein, which is an important component of the tubular structures in the axons and dendrites of neurons. PET scans are available to look at the amounts of plaques and tangles in the brain and are sometimes used to predict Alzheimer's disease, but are expensive and are most often used in Alzheimer's research. More than 40 clinical trials of drugs that remove beta amyloid plaques have failed to improve cognition, and some have worsened or accelerated the disease. Many other pathologies in the brain may occur during aging, which are also more exaggerated in the Alzheimer's brain. During middle age, the brain begins to shrink a bit each year, and this becomes very prominent in people with Alzheimer's disease. Therefore, even younger adults need to start thinking about prevention of dementia as they age. There is also deterioration of white matter with demyelination. In Alzheimer's disease, microglia and astrocytes become hyperreactive and secrete inflammatory substances, thereby producing neuroinflammation. Synapses are also damaged and lost. Abnormally large lipid droplets accumulate in ependymal cells around the ventricles, and studies have shown that enlarged lipid droplets may also be present in other glial cells. Insulin tends to accumulate within neurons that have neurofibrillary tangles. In Alzheimer's, there is suppression of neural stem cell proliferation and differentiation, insulin resistance with abnormal glucose uptake into the brain and brain cells, as well as mitochondrial dysfunction with oxidative stress and low ATP production can occur with aging, but is also much more exaggerated in Alzheimer's disease. We will focus in more detail on these last 2 points during this presentation. Cells, including brain cells, require fuel to produce the energy molecule adenosine triphosphate, or ATP, which is produced by way of the tricarboxylic acid cycle and electron transport within mitochondria. The brain is only about 2 or 3% of our total body weight, but the brain is an extremely active tissue even while we are sleeping and requires about 20 to 25% of the energy that humans consume. Neurons can contain 1,000 or more mitochondria to produce the energy that they require. This slide shows the difference between a normal glucose PET scan and a scan in the Alzheimer's brain. The yellow and red areas are areas of high glucose uptake into the brain, and it is obvious that there is much less glucose uptake in the Alzheimer brain. There are several factors that contribute to this, including insulin resistance and deficiency, mitochondrial dysfunction, pyruvate dehydrogenase complex 1 deficiency, and deficiencies in glucose 1 and 3 transporters, which allow glucose to enter the brain and neurons. It seems almost like there is a conspiracy of getting glucose into the brain. Without adequate fuel, the brain cells in Alzheimer's malfunction and may eventually die. This problem with glucose may be present for decades before symptoms appear. Glucose is the predominant fuel for the brain and other organs in people on a typical high-carbohydrate diet. However, glucose is not the only fuel we can use. Humans are programmed by evolution to switch to the use of alternative fuels during starvation when glucose stores have been used up. Glucose can be produced by way of gluconeogenesis from lactate and the amino acids from muscle breakdown. But humans would die within 1 to 2 weeks if we did not have fat. However, we can survive starvation much longer by using fat for fuel. When glucose stores are used up, fat in adipose tissue is broken down into fatty acids, which can fuel most organs but are not taken up readily into the brain. However, some of these fatty acids are converted to ketones in the liver, which are easily taken up by the brain and brain cells to use as fuel. Nutrition researchers in the lab of Dr. George Cahill Jr. first described the phenomenon in 1966 that during starvation, ketones provide about 2/3 of the fuel for the brain. The level of beta-hydroxybutyrate in particular becomes quite elevated within 10 days of starvation. Dr. Stephen Cunnane and his associates at Sherbrooke University in Canada have focused extensively on ketones, aging, and Alzheimer's disease using glucose and ketone PET imaging in adults of all ages, including cognitively healthy older adults, as well as those with mild cognitive impairment and Alzheimer's disease. Cunnane and group have shown that ketone uptake is normal in the same areas of the brain affected by Alzheimer's disease, which supports the idea of using ketones as an alternative fuel for the Alzheimer's brain. Cunnane has suggested that many neurons in the affected areas of the brain may be lying dormant and not dead as once believed, and are like cars with empty gas tanks waiting to be filled with fuel to run again. Using dual tracer ketone and glucose PET imaging in more than 300 young and older adults, Drs. Cunnane and associates have identified a brain energy gap in cognitively healthy older adults of 7 to 9% that worsens in mild cognitive impairment to 10% and further accelerates in mild Alzheimer's disease to 20% or greater. In one experiment, Cunnane and group used glucose and ketone PET imaging in people before and after 6 weeks of a ketogenic diet and showed that certain areas of the brain take up ketones preferentially when glucose and ketones are both available. Cunnane and group have also reported that the percentage of energy provided to the brain by ketones correlates directly with levels of ketones in the blood. In the BENEFIC trial of 65 people consuming C8-C10 MCT oil 15 grams twice daily for 6 months, Cunnane and group found that there is increased energy from ketones after consuming MCT oil and that 30 grams of MCT oil daily could provide about 5 to 10% of the energy requirement of the brain. The trial also showed that people with mild cognitive impairment consuming MCT oil had improved cognition in multiple domains, that ketones are taken up by white matter, and that the uptake of ketones in the dorsal attention network correlated with improvements in processing speed. This study also found a reduction in plasma cardiometabolic and inflammatory biomarkers and found no differences in results for people who did and did not have the ApoE4 gene. This slide shows a number of articles that summarize studies and mechanisms of coconut ketones and MCT oil for Alzheimer's disease. Where did the idea come from for using MCT oil for people with Alzheimer's disease? Oils are often separated into their fatty acids for various purposes. Until the late 1950s, C8 and C10, which are medium-chain triglycerides, were considered waste products of coconut oil. However, in the 1960s, intensive investigation revealed that there were therapeutic uses for these fatty acids, including treatment of malabsorption syndromes and use for oral and parenteral nutrition in infants through adults. In addition, coconut and MCT oil were added to infant formulas to provide medium-chain fatty acids found in human milk. In 1966, it was discovered in the lab of Dr. Theodore Van Italy and his associates that following consumption, medium-chain triglycerides are taken up by way of the portal vein directly to the liver and are partially converted to ketones. This will happen even if a meal with carbohydrates is consumed at the same time. Three ketones are produced when MCTs appear in the liver and are converted to acetyl coenzyme A. Ketones are also produced from long-chain fatty acids during fasting or starvation, or when a person consumes a high-fat, low-carbohydrate ketogenic diet, which has been used since 1921 to successfully eliminate or reduce seizures in people with drug-resistant epilepsy. MCTs are first converted to acetoacetate, which can then be converted to beta-hydroxybutyrate, which is the main ketone circulating in the blood. Acetoacetate can also be converted to acetone, which circulates in the blood but then is mostly exhaled. Ketones are small molecules that easily cross the blood-brain barrier and can provide fuel to brain cells as well as to other organs, except in the liver where they are made. This diagram illustrates that glucose and ketones can be taken up into mitochondria to produce ATP by way of the TCA cycle and electron transport. Glucose requires insulin either directly or indirectly to enter cells and eventually to enter the mitochondria, whereas ketones do not require insulin. When there is insulin resistance, glucose is not taken up normally into cells. However, ketones can bypass the problem of insulin resistance and enter the cell and mitochondria. Medium-chain triglycerides are not found in the most commonly consumed vegetable oils and fats, such as olive oil, soybean oil, canola oil, and even fish oil. However, coconut and palm kernel oil are the richest natural sources of MCTs. MCTs are also found in the milk fat of mammals, including humans, goats, and cows, and therefore MCTs are present in whole milk, cream, and full-fat cheeses. This is my hero, Dr. Richard Veitch, who worked on the TCA cycle during his PhD training at Oxford University with Dr. Hans Krebs, and then spent his entire career at the National Institutes of Health in the U.S. Dr. Veitch was a world expert on ketones until he died at age 84. Dr. Veitch took great interest in helping my husband, and I am very grateful for the many years he helped us. In the early 1990s, Dr. Veitch became interested in studying the possible therapeutic uses of ketones and was the first to develop an exogenous ketone supplement, a ketone monoester of beta-hydroxybutyrate and 1,3-butanediol, which can rapidly and substantially increase beta-hydroxybutyrate levels. Dr. Veitch's ketone monoester came on the market in the U.S. in 2018 and is also available in the U.K. The first experiments showed that beta-hydroxybutyrate improves the pumping efficiency of the heart while requiring less oxygen, and that ketones mimic many of the properties of insulin when glucose and insulin levels are low. Veitch and his associates also learned that ketones suppress the production of reactive oxygen species and act as scavengers of reactive oxygen species and are therefore anti-inflammatory. The first studies of ketones and brain by Dr. Veitch's group was published in 2000 and was an in vitro study of neurons that were subjected to either a toxin that causes Alzheimer's or one that causes Parkinson's disease. Cultures were incubated with beta-hydroxybutyrate or a control vehicle. Compared to controls, the neurons grown with beta-hydroxybutyrate survived, were larger, and had more outgrowths of neurites, suggesting that beta-hydroxybutyrate is neuroprotective and could provide a therapy for Alzheimer's and Parkinson's. As a result of the studies, Dr. Veach and others wrote several hypothesis papers between 2001 and 2004 proposing that ketone therapy could be beneficial in diseases that have decreased glucose uptake into cells, insulin resistance, and mitochondrial dysfunction, including the many disorders that are listed on the slide. Shortly after the Veach Group paper was published in 2000, Dr. Samuel Henderson patented the idea that ketones produced by consuming MCT oil might be adequate to improve cognition in Alzheimer's disease and developed a medical food that would be tested, leading to FDA recognition as a medical food. In a pilot study, 9 of 20 people had improved scores on cognitive testing after receiving a single 40-gram dose of C8 compared to another day on which they received a placebo. A second study of 152 people showed similar significant improvement at 45 and 90 days in people who were given C8 compared to controls. In both studies, people who were ApoE4 negative as a group improved on average, whereas those who were ApoE4 positive did not improve when their results were combined. However, an author later shared with me that some ApoE4 positive individuals did in fact improve. A press release about Dr. Henderson's studies gave me the idea to use coconut oil for my husband, Steve. My work as a neonatologist was very demanding with many emergency calls to the hospital. Steve voluntarily worked from home as an accountant for my medical practice, which allowed him to always be available for our children. Steve made it possible for me to be both a mother and a doctor, and I am deeply grateful to him for that. In 2001, at age 51, Steve began to have problems making payroll errors, missing tax deadlines, and then he couldn't remember if he'd been to the bank or post office. Steve had depression, and his doctor believed that his memory problems were related to depression. By 2004, his memory problems worsened, and he scored only 23 of 30 points on the mini mental status exam. Other causes were ruled out, and Steve was diagnosed with early onset Alzheimer's disease. Despite taking 2 Alzheimer's medications, Steve continued to worsen. By 2008, Steve was 58 years old and was declining rapidly. He had many of the symptoms, both physical and cognitive, that are shown on the slide. In May 2008, 2 new clinical trials for a vaccine and an oral medication that removed beta-amyloid plaque became available at Alzheimer's research centers near us, and Steve was scheduled at 2 different centers on 2 successive days. The evening before the first screening, I searched the internet for the risks and benefits of the 2 clinical trial drugs, and by chance came upon the press release for a new medical food called AC1202, which was not yet recognized by the FDA, which claimed to improve memory and cognition in nearly half of the people with Alzheimer's who received it. I found their patent application and learned that the only active ingredient in the medical food was the medium-chain triglyceride oil C8. The patent application also included information that was new to me, that Alzheimer's is a type of diabetes of the brain. In 2005 and 2008, Suzanne Delamonte and Mark Wand at Brown University in the U.S. reported that the brains of people with Alzheimer's who did not have type 1 or type 2 diabetes showed insulin deficiency and insulin resistance, and that this process is present in the earliest stages of Alzheimer's and worsens with each stage of the disease until it is widespread and severe. The patent application for AC1202 discussed ketones as an alternative fuel, the Veach Group studies of beta-hydroxybutyrate, and the Henderson Group studies of MCT oil. I recognized MCT oil because I had used the oil in the feedings of my tiniest premature patients in the 1980s. The patent application mentioned that MCT oil is extracted from coconut oil, which gave me the idea for how to help my husband, since I did not know whether MCT oil was readily available. Hours after I finished reading the patent application, Steve was screened for the vaccine clinical trial and scored a very disappointing 14 out of 30 points on the mini mental status exam and did not qualify for the study. Steve drew a clock a specific test for Alzheimer's disease, and you can see that he drew a few random circles and a few numbers that were not organized like a clock. We were advised that he was on the verge of severe Alzheimer's. We were very disappointed, and on the way home, I thought, what do we have to lose? And bought coconut oil at a health food store. When we returned home, I found the fatty acid composition of coconut oil on a USDA website. I learned that coconut oil contains about 60% medium-chain fatty acids with chain links from C6 to C12, and estimated that about 35 grams of coconut oil would be equal to the 20-gram dose used in the second clinical trial of the medical food AC1202. The following morning, Steve received 35 grams of coconut oil with his breakfast and warm oatmeal. About 4 hours later, during the second clinical trial screening, Steve scored 18 out of 30 points on the mini mental status exam, 4 points better than the day before, and this time he qualified to be in the clinical trial. Every morning after that, I gave Steve 35 grams of coconut oil for breakfast. However, I had read in the patent application that the ketone levels after taking MCT oil peak at about 90 minutes and the ketones are back to baseline at 3 hours. Since the brain needs fuel 24 hours a day, I began to add coconut oil to the other meals. Steve had many improvements during just the first few days, and here is a visual example. As a reminder, the slide showed the clock that Steve drew the day before he started consuming coconut oil. This is a clock that Steve drew 14 days after starting coconut oil. You can see that it is messy but much more organized. There is now one large circle and all 12 numbers are present. There are many extra lines, and Steve said he drew the lines to try to place the numbers across from each other on the clock. This is the clock that Steve drew 37 days after starting coconut oil. It is tidier and there are not as many extra lines on the clock. Steve said he felt like a light bulb came on in his brain the day that he started consuming coconut oil. This slide shows the many improvements that Steve experienced during the first days, weeks, and months. Some of the most significant were that by 2 months he was able to tie his shoes again and his gait was now normal and he could run again. Steve had not been able to read for about 1.5 years, but at 3.5 months after starting coconut oil, he was able to read again. He explained that the words would shake around on the page when he was trying to read and that the shaking had stopped. About 1 week after Steve started consuming coconut oil, I began to telephone ketone expert Dr. Richard Veach, who did not believe the ketone levels would be high enough from consuming coconut or MCT oil to help someone with Alzheimer's disease. He had shared with me that he was developing a ketone monoester that could increase ketone levels 10 times higher. At 2 weeks, I then faxed the copies of Steve's first 2 clocks to Dr. Veach, who was quite surprised, and he sent me his hypothesis papers, other studies, and he connected me with other ketone researchers. Several weeks later, Dr. Veach measured Steve's ketone levels before and for 3 hours after taking 35 grams of coconut oil at breakfast and then at dinner. The slide showed that acetoacetate levels were higher than beta-hydroxybutyrate levels and reached a peak of about 0.35 millimoles of total ketones after the second dose, although the levels still appeared to be increasing 3 hours after that dose. About 2 weeks later, Dr. Veach suggested measuring ketone levels again after taking a 20-gram dose of C8 MCT oil in the morning, along with a second subject. Steve's levels are shown in black on the slide. After the MCT oil, beta-hydroxybutyrate levels were higher than the acetoacetate levels, and total ketones peaked at about 1.5 hours at 0.45 millimoles. Thereafter, I began to provide Steve with both coconut and MCT oil. The amount of MCT oil Steve was consuming was gradually increased. He did not tolerate much C8, therefore I used a C8-C10 MCT oil and experimented with mixing the oil with virgin coconut oil. The rationale for the mixture was to obtain higher levels of ketones provided by MCT, but more prolonged levels provided by coconut oil, along with other beneficial fats and substances in coconut oil, such as lauric acid, which I had learned was antimicrobial. I believe that the mixture might help Steve sustain nearly constant ketosis. I found that Steve tolerated a ratio of MCT to coconut oil of 4 to 3, and the oils in the mixture did not separate, and the mixture remained liquid at room temperature. The mixture blended very well into many cold and warm foods. Over several months, Steve's intake of the oils was slowly increased to 45 grams 3 times a day with his meals, and then 30 grams before bedtime, which seemed to help him sleep better. Steve consumed much less fruit, bread, pasta, and other carbohydrates and was effectively on a ketogenic diet. Steve tried again for the first clinical trial and scored 20 points on the MMSE. He had his choice of which clinical trial to enter, and we chose an oral medication called semaglutide, which was a crossover trial. We advised the director of the research institute that Steve was taking coconut and MCT oil, and the doctor was familiar with this idea because the group had participated in the AC-1202 clinical trial and allowed Steve to participate in this trial. We were later advised that Steve was on the placebo for 18 months. Therefore, the improvements he experienced during that time were almost certainly related to taking coconut and MCT oil and the ketogenic diet. During the first year of the clinical trial, Steve had improvements in his scores by 6 of 75 points on the Alzheimer's disease assessment scale cognitive and by 14 of 78 points on the activities of daily living test. At the time of the Alzheimer's diagnosis in 2004, Steve's MRI was reported as normal. However, in 2008, when he entered the clinical trial, there was moderate to severe atrophy, most obvious in the hippocampus and amygdala. 2 years after starting coconut oil in 2010, there was no increase in atrophy on the MRI. We now know that Steve crossed over to semaglutide in January of 2010, and within 6 weeks had a change in his hair color along with many adverse effects shown on the slide, as well as the return of some of his previous symptoms, but also new symptoms of Alzheimer's disease. Steve withdrew from the study, and we later learned that semaglutide was found to accelerate Alzheimer's disease. I was in frequent contact with Dr. Veach, and toxicity had been completed on the ketone monoester with minimal adverse effects reported. Dr. Veach suggested a pilot study of the ketone monoester in which Steve would be the only participant. Steve began with 22 grams of ketone monoester 3 times a day. Within 2 hours of the first dose, Steve's mood improved and he could sing and write out the entire alphabet again. Before receiving the ester, Steve required step-by-step assistance to take a shower, shave, and brush his teeth. The day after the first dose, Steve was able to take a shower, shave, and brush his teeth without assistance, and we saw complete reversal of the new and old symptoms within several days to 6 weeks of starting the ketone monoester. Steve told me that he felt good, he had more energy, was happier, and that it was easier for him to do things, which I agreed with based on my observations. Steve remained stable for about 20 more months before having another setback in the disease. Dr. Veach believed that we should aim for beta-hydroxybutyrate levels of 4 to 5 millimoles, which on testing was achieved with a dosage of about 25 grams, which Steve received 3 times daily thereafter. In early 2015, I was pleased to be the first author on a case report published in the journal Alzheimer's and Dementia, which detailed Steve's dramatic improvement while taking coconut and MCT oil over a 2-year period, as well as his improvement and stabilization for 20 more months while taking Dr. Veach's ketone monoester. Sadly, in 2013, Steve had fallen and hit his head due to a generalized seizure lasting 20 minutes with apnea and cyanosis and became completely dependent thereafter. He continued to receive ketogenic therapies. However, Steve passed away on January 2nd of 2016. Steve donated his brain, and his autopsy report showed that he had passed away from advanced Alzheimer's disease as well as Lewy body dementia, which has many symptoms of Parkinson's disease. Now I would like to tell you about a collection of reports about other people with memory impairment and their responses to taking coconut and MCT oil. Back to 2008, I believe that since Steve had improved so much while taking coconut oil, Other people with Alzheimer's would also improve, and it became my mission to try to get the message out. I wrote to politicians, the media, and many others, including the Alzheimer's Association, telling them about AC-1202, and I wrote a white paper about Steve's improvement and the urgent need to study coconut oil, MCT oil, and especially Dr. Veitch's ketone ester, which had not yet received funding for studies of Alzheimer's from the NIH. A newspaper article went viral, and not long after, I began to receive reports, mostly from caregivers, about the responses of people with Alzheimer's and other memory impairments to taking coconut oil and/or MCT oil. After accumulating about 40 reports, I began to enter the data on an Excel spreadsheet, since the information could be helpful in acquiring funding for studies. The writers were not prompted as to what improvements they might expect, but if they reported in simple terms that the person improved, I would ask them to provide more details. Between late 2008 and mid-2014, I had collected 360 anecdotal reports, of which 288 were about people who had memory impairment related to dementia or another disorder. I finally had an opportunity to present these anecdotal case reports in a poster presentation at the Alzheimer's Association International Conference in Amsterdam in July of 2023. In April of 2024, I had the opportunity to publish this study, along with information about ketones and medium-chain fatty acids, in a special Alzheimer's issue of Medical Research Archives. I do not consider this a scientific study, since I believe more people would write to me if they had a positive response than if they had no response. However, many people reported that they saw no improvement. I tabulated from the reports how many people reported improvement, no improvement, or stabilization for at least 3 months, as well as what types of improvements they reported. Verbatim phrases from the emails, as shown on the slide, were used to further categorize the types of improvements reported. The ages range from 33 to 94 years, with slightly more females than males, and the breakdown of diagnoses and other conditions reported are shown on the slide. Altogether, there were 288 reports, with Steve Newport as the index case. Of the 288 people, 72% consumed only coconut oil, 26% coconut and MCT oil, and 4% MCT oil only. Overall, 89.2% reported improvement, 7.3% no improvement, and 2.4% no improvement but stabilization for at least 3 months. Many people reported more than one improvement, and you can see on the slide that about 2/3 of the people reported improvements in memory and cognition, about 1/3 reported improvements in their speech, language, and verbal skills, 1/3 reported improvements in social interaction, behavior, or mood, about 1/4 reported that they had been able to resume self-care or other activities, and almost 20% reported improvement in physical symptoms, as well as a small percent reporting improvements in sleep, appetite, or vision. Some people have stayed in touch and have reported sustained improvement for up to 10 years. This shows that there are other types of symptoms involved in Alzheimer's disease beyond memory and cognition that could improve by consuming coconut or MCT oil. Improvement in symptoms, and especially in the ability to take care of one's daily needs, can be very meaningful for people affected by memory impairment, as well as their families and caregivers. We will now discuss some other factors related to ketones, coconut, and MCTs that could be beneficial to the brain. In addition to providing ketones as fuel to the brain, beta-hydroxybutyrate from consuming MCT oil has been shown by several groups of investigators to reduce brain inflammation by way of inhibiting the NLRP3 inflammasome and by acting as an antioxidant to reduce production of reactive oxygen species and by acting as scavengers of free radicals. Beta-hydroxybutyrate was also shown to prevent perforation of neurons by beta amyloid in an in vitro study by Yin and others. Consumption of beta-hydroxybutyrate was also shown to reduce the amount of beta amyloid plaques and neurofibrillary tangles in the brain, decrease anxiety, and preserve cognition compared to controls in studies of Alzheimer's mouse models conducted by Dr. Veitch's group, and the study was confirmed separately by Wu and others. In the U.S., there are various MCT oil products now available, including C8 alone or various mixtures of C8, C10, and C12, along with Dr. Veitch's ketone monoester and other types of ketone esters, ketone salts, and mixtures. Coconut oil contains about half C12 and 12 to 15% of C6 to C10 combined. Conversion to ketones is most rapid for C6 and C8, followed by C10, and hours later by C12. Even though the peak ketone level is lower for coconut oil than for MCT, the slow conversion to ketones could make ketones more available continuously to the brain. C8 and C10 are known to cross the blood-brain barrier and to directly fuel the TCA cycle in astrocytes, and therefore these medium-chain fatty acids can also provide an alternative fuel for the brain. It is not yet certain whether C12 crosses the blood-brain barrier, and hopefully such a study will be completed soon. C10 has been shown to promote multiplication of mitochondria, which would increase the availability of ATP, as well as thousands of proteins produced in mitochondria. C10 also has an anticonvulsant activity that is equally effective to the anticonvulsant medication valproic acid. A group in the UK has developed an 80% C10 MCT oil called K-Vita to treat people with epilepsy. A study was performed by a group in Japan to understand more about how coconut oil might have helped my husband Steve despite relatively low blood levels of ketones. In a study of mice, levels of C12 after consuming coconut oil were found to be quite high and to persist for hours longer than levels of C8 and C10 after consuming MCT oil. C8, C12, and C18, a long-chain fat that does not cross the blood-brain barrier, were also found to stimulate ketone production directly in vitro in KT5 mouse astrocytes. The results of these two experiments suggested that C12 could be more available to the brain because of the higher peak levels and much longer presence of C12 in the blood. If this is proven in vivo and C12 is confirmed across the blood-brain barrier, this suggests that C12 may have a more direct ketogenic effect within the brain. Lauric acid is also known to be highly antimicrobial to a variety of bacteria, viruses, fungi, spirochetes, and protozoa. C12 has been shown to kill many of the microorganisms such as herpes simplex that have been implicated in hundreds of studies as possible causes or contributor to Alzheimer's disease. Lauric acid represents about 6% of the fat in human breast milk and is known to help protect the newborn from infection. There are many commercial applications for lauric acid and monolaurin as antimicrobial agents in medical and veterinary applications and for uses in oral care products, soaps, and disinfectants. In Alzheimer's, activated microglia release nitric oxide and pro-inflammatory cytokines, which promote increases in the other neuropathologies like plaques, tangles, and insulin resistance. Nishimura et al. Studied activated mouse microglia and found that exposure to C12 in vitro suppressed the production of nitric oxide, reactive oxygen species, and pro-inflammatory cytokines and suppressed phagocytosis. Ischemic stroke increases blood glucose acutely and produces toxic glucose metabolites with poor utilization of glucose as an energy source, as well as oxidative stress, especially in small blood vessels. In a mouse model of ischemic stroke with elevated blood glucose, Shahivar and others found that the presence of C12 increased the diameter of blood vessels, promoted the growth of new blood vessels, and stabilized barrier functions, which reduced the extent of damage in the brain. A study by Carenti et al. Found that middle-aged and older people in Italy who had moderate consumption of MCTs, especially C12 and short-chain fatty acids, were less likely to have cognitive impairment. They also reported that people with low blood levels of acylcarnitines containing medium-chain fatty acids, including C12 and the short-chain fatty acid 2-hydroxybutyric acid, were more likely to have Alzheimer's and a lower volume of prefrontal gray matter. In 2020, Dr. Ralph Martins of Australia and his group published a review of the potential of coconut oil for prevention and treatment of Alzheimer's disease. In addition to the ketogenic effect, they reported that virgin coconut oil is also rich in anti-inflammatory and antioxidant polyphenols, and that a substance in virgin coconut oil was shown to protect survival of neurons and suppress mitochondrial alterations in neurons exposed to beta amyloid. Fernando and others have also reported that people with the ApoE4 gene consuming coconut oil were found to have improved cognition on the mini mental status exam compared to people who are ApoE4 negative, and the people in another group consuming canola oil. In addition, both oil groups had average reductions in total and LDL cholesterol. However, the people with the ApoE4 gene consuming coconut oil had larger reductions in their total and LDL cholesterol levels. This is the opposite of what was found in Dr. Samuel Henderson's MCT oil studies. The reason for this difference is not yet known. For decades, dietary guidelines have advised avoiding consumption of tropical oils due to their high saturated fat content. Coconut oil is high in saturated fat, but about 2/3 of the saturated fatty acids are the medium-chain fatty acids, shown in light and dark blue on the slide, which are digested and absorbed differently and have different biological properties than long-chain fatty acids. Of the oils and fats shown, only milk fat contains significant amounts of MCTs. A 2020 review of studies comparing coconut to other oils from 1985 to 2019 reported that coconut oil increases LDL cholesterol. However, 6 of the 16 studies showed average decreases in LDL cholesterol, and 15 of 16 studies showed average increases in beneficial HDL cholesterol. In a review to be submitted soon for publication of studies lasting for 3 weeks to 2 years, there were 525 data sets for differences in lipid profile values for people consuming coconut oil. When all data were combined, the increases in total and LDL cholesterol were less than 0.8%. When broken down into short, medium, and long-duration studies, only the short-term studies showed average increases in total and LDL cholesterol of less than 5%. These results suggest that the long-held belief that coconut oil increases cholesterol is incorrect, and that if LDL cholesterol increases at all, this effect may be temporary. Therefore, an individual may experience an increase, decrease, or no change in lipid values in response to consuming a new oil during the early weeks. Doctors and people with diabetes often worry that taking coconut or MCT oil could cause diabetic ketoacidosis, but this is extremely unlikely because the ketone levels are many times lower than those that occur in diabetic ketoacidosis when insulin is not available and blood glucose levels are extremely high. However, it may be possible to develop temporary ketoacidosis not due to diabetes from consuming too much ketone ester or too many high doses close together, but the levels should come down spontaneously and very quickly. You can obtain a list of references for coconut oil, MCT oil, and ketones for Alzheimer's disease by scanning the QR code and looking for the first entry on that page of my website, coconutketones.com. You can also contact me by email as shown on the slide. Thank you so much for your kind attention to my presentation.